Hello everyone, and welcome to today's webinar. My name is Caitlin McCann, and I will be your moderator. This webinar is titled Cosmic, Combining Expert Curation with Scientific Innovation in a Gold Standard Database for Precision Oncology. This presentation is brought to you by QIAGEN Digital Insights. I'd like to now welcome our speakers, Dr. Zbyslaw Sondka, Senior Scientist, and Rebecca White, Scientific Communications Manager, both with Cosmic at the Wellcome Sanger Institute. And with that, I'll hand it off to Rebecca.
So I'm going to kick off with just a bit of a description of why Cosmic, why Cosmic exists, what we do, and how we do it. And then I'm going to hand over to Zbyslaw to talk about a bit more about our products and how you can get the most value from them. So starting with the why. We exist to accelerate the advancement of cancer research, be that diagnosis, treatment, or prevention. And ultimately, we do that to improve the length and quality of life for all cancer patients. Our vision is that one day a patient will go into a doctor's surgery, and as soon as they hear a doctor say to them, "I'm sorry, you have cancer," it's followed up immediately by the words, "But it's okay. We know exactly what to do about this and exactly what your treatment journey will be." What do we do within that context? Well, we collate, streamline, and quality filter global cancer somatic mutation data. We make that data useful and easy to understand, and we draw out salient insights. The idea is that our database will accelerate research without compromising on quality. And exactly how we do that is through expert curation, data interpretation, and visualization. We're experts in identifying data, extracting it, standardizing it, and presenting it. Now, I'll go into a bit more detail on this later, so don't worry about the notes here. And I just wanted to kind of start with that why and positioning ourselves within cancer research. So as many of you will know, cancers are no longer thought of as one single disease. It can be thought of as a group of diseases that share common characteristics that give them an advantage and abnormal behaviours that help them to grow and thrive. And these are really nicely described through the hallmarks of cancer. Excitingly, the hallmarks also represent really good strategies for tackling cancers. If you can take away things like their ability to grow vasculature and their food sources, if you could get immune cells to recognise the cancers, or if you could somehow stop the cancers from growing and spreading, that would be a really good way of stopping things. But it's a bit more complicated. We know that obviously there's not one single mechanism by which tumours and cancers achieve these things, and they do it in multiple different ways. And sometimes even if you tackle one aspect, there might be a redundant mechanism where cancers are doing it in multiple ways. And I'll come into more on this later and why it's important in the context of somatic mutations. And of course, we also need to think about cancer on lots of different levels of biological organisation in terms of the environment and what sort of mutagens are exposed to the body and how all of our bodies are slightly different, through to the actual environment, microenvironment and specific tissue differences, to the cellular level and what's going on there, to the proteins and their dysfunction and dysregulation, and finally down to somatic mutations or mutations in the genes. And all of these changes participate in the development of the disease, but it starts with our genes. And that's why somatic mutations are so important. They help us to understand the bigger picture. Through analysis of somatic mutations, we're able to link DNA change to protein dysfunction. We're then able to associate those known functions of the protein with biological processes and identifying the pathways and processes involved in disease progression. Again, this is important because if we think back to the fact that you can get resistance or certain treatments in isolation don't work, that's because a few mutations, if you look at them, might be impacting multiple pathways, which give the cancer the same ability to maybe grow uncontrollably. So you need to think about how you can target multiple pathways. And then choose an optimal therapy regime or propose a new therapy regime, or in fact, tell a patient we know this treatment won't work, saving months of unnecessary treatments. And finally, it's unpicking the etiology of cancer. If you can combine mutations with information about things like environment, you can begin to understand what's causing cancers for individuals. Now, so in the ideal world, what we have is genetic information linked to the cancer information, linked to treatment information. But again, another challenge is there's just too much data and there's not enough time. In 2020 alone, there were more than 30,000 publications that linked cancer and mutations. And that's just way too much for any single person to keep up to date with. And they're still missing information. We don't know exactly which mutations are driving cancer, the functions of the products of those mutations, or which mutations could be good candidates for drug development. We know some of it, but not all of it. And we also end up working in niches and focusing on, say, a specific cancer type or a specific protein type. So what you need is a big database that collates all of this so you can begin to see the patterns across cancer types, across mutation types, and how maybe one treatment may be applicable in lots of different types of these diseases. So as I said, that data becomes useful once collated, standardized, and structured so that insights can be extracted. And that's exactly what Cosmic does and what we're experts at. And in fact, we've been experts for 20 years. We started out in 2002 at the Sanger Institute. That's before the Human Genome Project was even completed. It's a spreadsheet of the Sanger Institute's Cancer Genome Project to keep track of genetic mutations. And it started out as just 4 genes, BRAF, HRAS, KRAS, and NRAS. just 67,000 samples and 10,000 mutations. It was so useful, it's been grown and it's sustained. To today, we include all genes, more than 1.5 million samples, and 23.4 million mutations are recorded. Our data resolution is unparalleled, and our experience is unparalleled, and that's why we are the gold standard database. So how exactly do we go about doing this? Well, this is kind of a broad summary. We gather data from high quality and trusted resources worldwide. Mainly, this is scientific literature, but we also get data from some of the wider cancer genome consortiums and data from some smaller studies. And more recently, we've also been getting data from clinicaltrials.gov and other clinical trial resources in our actionability product. We combine all this data and we run it through a large standardization and quality control pipeline and map it to other existing resources and use those to annotate our data, such as Ensemble. And then that is then produced into a range of products. The most common one is our download file, but then we also have a suite of data visualization and data interpretation tools, which will be gone into in more detail later. But I'm going to spend a bit more time focusing on where our data comes from. So we have two different types. We have this broad genomic data, and that's, as I said, comes from these from semi-automation annotation of the big genome screens, and also genome-wide studies that are published in the scientific literature. We take the supplementary data sheets and run them through genetic annotation systems, and then all of that is mapped to both Human Genome Assembly 37 and 38, and you can toggle between both on our website or download data on both. But our real kind of area of expertise is in manual curation. I said earlier that we were first formed in 2002, and we still have some of that curation expertise from when we were first formed in the team and being passed down. Our curators are postdoc level experts who they have brilliant expertise in being able to extract data and high quality data from publications. And it's not just the gene data, they also extract data on things like cancer type and stage, and also some kind of wider clinical metadata around sex, age, family history, prior treatments, certain environmental factors, as well as the mutation status and sample source. And this deep genetic data isn't random. It actually comes from our cancer gene census. So the cancer gene census is currently 729 genes which are known to have to be drivers of cancer. And that's the literature proves that they have a mechanistic role in driving cancer forward. There's tier 1 and tier 2 genes. Again, this will be discussed in a little bit. But those genes are where we focus our gene curation because we know they are the relevant genes and it removes some of the noise of irrelevant things. But we also have disease-specific curation. So each release, our curators will focus on a specific disease, and usually this is rare cancers. Now, what's interesting about the disease curation is through reading all those papers, our curators identify new genes that should be put forward as candidates for the cancer gene census because they're the role in cancer they're seeing in these papers. And then that gets handed over to our cancer gene census expert who will then validate or invalidate and then add that gene either as tier 1 or tier 2 to our census. So in our last release, V96 in May 2022, we have a focus on head and neck cancers, which account for 3 and 5% of the US and European cancer incidence. Rather than focusing broadly on all head and neck cancers, they've done a specific focus on the rarer forms of head and neck cancer, those that affect salivary glands, sinuses, or muscle and bone in the head and neck. That data has come from 56 publications. And this is a really nice example where PRKD1, a serine threonine kinase, was added to the cancer gene census as a tier 2 gene because it was found to have a role in cribriform adenocarcinomas. So this is just to show how all of our data links together. And then the end outcome in terms of pure stats is what you're seeing here. We have a total of currently 23 million mutations from 1.5 million samples, 28,000 papers, and 39,000 WGS samples. And we also record things like copy number variants, fusions, differentially methylated CPGs, and gene expression variants. So now I'm going to hand over for a more detailed look at our products and how you can use them.
Thank you very much, Rebecca. There are many types of cancer. They originate from different tissues, and it's hardly a surprise that they are different on the tissue level, but they are also different on genetic level. And Cosmic can help you with finding out what are the molecular differences between these cancer types. So when we take the lung cancer, 85 of all lung cancers are non-small cell lung carcinomas. They are rarely diagnosed before they become metastatic, and this is a major cause of cancer mortality. By analyzing cosmic data, we can identify very easily top 3 mutated genes in that form of cancer, and these 3 genes very nicely illustrate some somatic cancer genetics. KRAS is a very good example of dominant oncogene. What you see on this graph are mutations from all cancer samples with mutations in KRAS, presented all together on one histogram. So the Y scale presents the number of observed mutations, while the X scale presents the length of the gene. And you can immediately see that majority of mutations in that gene are focused around one very discrete peak of recurrent mutations. This pattern straight away suggests us the gain of function of activity. And in cancer, you can have 2 types of genes. One of them is oncogene, where you expect the gain of function. So the protein is hyperactive, and this hyperactivity drives cancer. The other major category are tumor suppressor genes, where the mutation causes loss of function. So the protein cannot act normally and doesn't protect the cell from oncogenic transformation anymore. As you can imagine, it's much easier to break something than to make it working more efficiently. And this is exactly what we see here. So it requires very precise change to make KRAS more active. And this is also confirmed by other types of alterations that are observed in cancer. You can see on the bottom of this graph, you have a gain of copy number variants. That means that this gene is frequently amplified in cancer cells and also is frequently overexpressed. When we take a closer look at this cluster of mutations, you can see that this is mostly codon 12 that is affected. And if we see the change on the DNA level, also not all the components of the codon, not all the nucleotides that compose the codon are affected equally. And this is mainly due to 2 factors. The first one are the constraints put by genetic code, because a particular amino acid change is required to actually increase the activity of KRAS. And the other reason is the type of the factor that actually causes the mutation. And you can see a very nice example here. In lung, the majority of changes on the nucleotide level are guanine to thymidine, and this is because that's how tobacco smoke mutates DNA. In colorectal carcinoma, KRAS is also a known driver, but the composition of mutations across cancer samples is slightly different. Here you can see the dominance of G2A substitutions, and this is likely caused by eating too much of burnt meats like smoked bacon and heterocyclic amines that are included in that diet. I'll get back to this graph later, but let's get back to another gene. Let's get to another gene, TP53. This is a classic tumor suppressor gene. You can immediately see the difference in the distribution of mutations across this gene. It's more even. Still, there are some peaks of recurrent mutations, and this is another thing I will get back to later. But what is also different are these 2 tracks that show complex and insertions and deletions. Insertions and deletions in this case often result in nonsense or frame shift consequences, and this causes loss of function because after such mutation the protein change is not synthesized anymore. So as a result, the protein is inactive and it cannot protect the cell from tumor. The third gene, most frequently mutated in lung cancer, is EGFR. This is another oncogene, but the pattern of mutations is slightly different here. So you can see these three peaks of substitutions here, similar to what you observe in KRAS. But also insertions and deletions track also contains many recurrent mutations. What is different compared to TP53 here is that these insertions and deletions are mostly in frame. So that means that only tiny bit of a gene is either removed or added, but this tiny change results in gain of function. So basically, EGFR is a transmembrane protein that accepts signaling from outside of the cell, and it passes this signal down to the cell, which results in increased proliferation and other consequences. When you have one of these mutations, the protein seems to be active all the time, and this activity is not dependent on external stimulus. So no matter what the tissue needs from this malignant cell, the cell is doing its own program instead of doing what's good for the body. That's how cancer starts. Now we have these in-frame deletions in EGFR, and again, their function is destroying the control over activity of the protein. So mutant forms are always on, and the control of the body over the proliferation of these cells is limited. What's important in these different mutations is that there are different drugs that are designed specifically to address them. So knowing which mutation is present in particular cancer sample gives you the information what drug should be used in this case. Another interesting mutation in EGFR is, as you can see, recurrent substitution T790M, so-called gatekeeper mutation, and it's very interesting because it causes lung cancer resistance to certain drugs, and it often arises as a result of gefitinib monotherapy. So once you detect this mutation, it's already clear that this particular drug won't be efficient to help this patient. What's interesting, this mutation is not cancer promoting on its own. You can see it arising very often as a result of the therapy, but once the drug is removed, then mutation disappears. So again, information about presence of this mutation can be used to precisely modulate the therapeutical regime. So that was the high resolution on the genetic level. COSMIC also provides high resolution on the level of phenotype. So in other words, different forms of cancer. You can select the exact form of cancer, and you can see, like I showed you for the lung cancer, you can see for each cancer type which genes are most frequently mutated there. So for example, if we take hematologic neoplasms, we can compare them against each other, which genes are predominantly mutated here, and you can see typical blood cancers, AML, myelodysplastic syndrome. They have different drivers, and that means that they possibly can have different targets as well. You can also go deeper. So if we compare just myeloproliferative neoplasms, we can see several subtypes, and many of the genes that drive these subtypes are the same, with exception of mast cell neoplasm, which is very rare form, and it is driven predominantly by TEAD. And this is where really interesting things happen. When you combine this phenotypic resolution and genomic resolution together, you can see how these two different diseases, even though driven by the same gene, are different on the genetic level. In mast cell neoplasm, KIT is mutated mostly within the phosphotransferase domain, and it is represented mostly by the H16V substitution. On the other hand, in gastrointestinal stromal tumor, again, KIT is a driver of this disease, but the mutations are focused mostly around juxtamembrane domain. So this is a result of differences in biology between these two tissues. The effect is the same. The tumor is developing thanks to these mutations, but slightly different changes to function of the same protein are needed for the tumor to develop. And this information is quite important when you are working on variants of unknown significance, when you are trying to discover specific biomarker, or when you try to select the right pharma targets. Because again, you can design a drug against the gene, but in fact, this may not be targeting the mutation that is important for this particular disease. Fusions between two or sometimes more genes are also major driving mechanisms for cancer, and very well-known examples are BCR-ABL1. This is most common fusion in cosmic as well. It drives leukemia. These fusions are manually curated. In cosmic, they have separate resource. At the moment, we have 305 gene pairs, but that means much more fusions because we present them in terms of exon connections. This is the highest resolution we can get in most cases because it's RNA-seq that is mostly used to detect these mutations, so we don't have precise intronic information if the breakpoint is there. But if we have information about exact genomic coordinates of the breakpoint, we record that too. So this is cosmic. How all these millions of mutations can be seen in a very simple way, and that already gives you some valuable information. So it allows you to identify mutations and genes typical for each cancer type. It allows you to see which genes are involved in development of particular cancer type. And to a certain degree, by looking at the patterns of mutation distribution, you can predict if the impact of mutations is activating or deactivating. Then you can find some clues about the origin of mutations. So if it's caused by the smoke, by the tobacco smoke, or maybe exposure to wrong food. But there are many other important questions that are needed to properly help cancer patients. For example, which of the thousands of genes and millions of mutations drive cancer and why they do so? So we have typically in every cancer samples thousands of mutations, and only tiny fraction of them are actually not the result of the processes that drive cancer, but the cause of cancer progression. Then even smaller fraction of these driving mutations are actually actionable. So we would like to know which mutations create therapeutic opportunities and which make therapy ineffective. Another question is how we can better determine what mechanisms drive genomic instability. I showed you this example with tobacco smoke and burnt meats. But this is very simple and we can do much more. And in the second part of the presentation, I'll tell you more about it. And I would like to show you several resources that actually help interpreting these data about cancers and mutations gathered in Cosmic database. Cancer gene census, this is our oldest resource. It's manually curated. At the moment, it includes 729 genes. It started with just 4. We curate it manually, and before qualifying a gene to this resource, we are looking for very convincing evidence that this gene is actually functionally involved in cancer progression. In addition to that, we have functional descriptions of how each gene contributes to cancer. So, for example, all these informations about what particular mutations in EGFR are doing, what is the mechanism of carcinogenesis through these mutations, you can find in these functional descriptions. So we categorize different functions of this gene that are relevant for cancer development in terms of 10 classic hallmarks of cancer. And you can see very brief descriptions of what this gene does to drive cancer through any of these biological functions. And you can also find links that will direct you to PubMed, and you can read the whole papers where this information is coming from. COSMIC is best known for its collection of somatic mutations, but of course, it's not only about mutations and gene fusions when we are talking about driving cancer. These cancer-driving genes can be altered in many different ways as well. And here is a good example of the APC gene. This is a classic tumor suppressor gene known to drive colorectal cancers, and you can see that many samples in COSMIC have this mutation. But in case of ovarian cancer, the number of mutated samples is actually not that big. Instead, this gene is dysregulated on the gene expression level. So protein is not expressed and cannot work. So there is no mutation, but other type of alteration that gives the same result and drives cancer. So you can see different types of this alteration all together and identify cancer drivers not only on the DNA level, but also on the regulation level. And this is all about how cancer happens. But then very important question is, if we understand what's happening in the malignant cell, is there anything we can do about it? And to address this problem, we have another manually curated resource, COSMIC Actionability, that aims to capture the current state of precision oncology. So for each mutation and cancer type, we have information about current or past clinical trials, including their results, availability of approved or tested drugs, and that includes the whole journey of the drug through clinical trials. So we use several ranking levels. The first one is already drugs that are already approved and on the market. Then the second level are drugs in the late phases of the clinical trials that already have met primary objectives, so very promising one and with very high probability of being approved. But we also have the drugs that are being tested on earlier levels that are looking promising. And finally, we also have case studies, and this is where the power of manual curation can be seen, because I'm sure you can see the news about new cancer drugs, new promising cancer drugs every week. But then when you read them, it occurs that they are just in vitro, very initial tests. And most of these cases you never hear about again. So in our case, curators are selecting the trials that are particularly interesting and that they feel the community should know about. Then we have some remarks on the progression. So we have certain trials that are cancelled, but not all of them are cancelled due to serious adverse effects. If they are, we make a note about that, but some of them are cancelled for different reasons. For example, the company is not interested in oncology anymore, so they just cut the funding. But still, this drug can be potentially used against the cancer it was tested for or against some similar cancer. So that allows our users to find these very promising candidates and decide what they would like to do with it. As I said, this is manually curated resource, and it's not as simple as just going to the clinical trial database records. One of our early discoveries was how incomplete, surprisingly incomplete these records are, and more than half of our evidence in this resource are actually coming from literature, from articles, from conference proceedings, from producer websites, and from places like FDA website. We don't use untrusted sources. We are very conservative about that. But this additional curation actually brings a lot of value in terms of the results being up to date and comprehensive. Actionability has separate release cycle because the priority for us is to be for it to be as up to date as possible. So it's being updated 4 times a year, and it's being expanded with the new genes and with new cases of the trials for the genes that we already have. So currently, there are 49 genes curated completely, so the full information about these genes is available there, with 235 other genes partially curated, and we are expanding this continuously. T790M mutation from EGFR was an example of mutation that brings resistance to certain therapies. And there are more mutations like that, and we do curate them as well. So it's not only about this one case. We have separate resource that tells you that this particular mutation causes resistance to these particular drugs. In this case, you have a list of mutations for imatinib together with the number of samples that these mutations were detected in. This is just the top fragment of that list. It goes well down to the floor, and you can use that resource as well to identify the mutations you need to be aware of during the therapy. That if they exist, you need to modify the approach to cancer. Now, the problem that I was mentioning, thousands of mutations in cancer, only some of them are really doing something for the disease progression. Cancer mutation census is our approach to prioritize mutations which actually are causing protein dysfunction. So this is an example of NRAS, a close cousin of KRAS mentioned before. And you can see here, again, the length of the gene and different mutations in cosmic and which class they are. We have a classifying system that assesses the likelihood of mutation being a driver of the disease. And we do that by applying some rules and integrating multiple resources, including CleanVars. So we have this manually curated component here, which is very high certainty. We have, of course, integrated the cancer gene census as another manually curated resource. We use patterns of somatic mutations. So, for example, if we know this is an oncogene, we prioritize mutations that are recurrent within these distinct peaks. If it's tumor suppressor gene, we are prioritizing nonsense or frameshift mutations. But we also combined some algorithms to help that. And here we use DNDS, which is a measure of selective pressure acting on a gene in particular cancer. Basically, that means that if this mutation occurs in this cancer, you can say it's good for this cancer. because it's preferred in the cancer cell population. On the other hand, some of the known somatic variants are identical to the germline variants, the single nucleotide polymorphisms found across human population. So to eliminate these cases, because if they are frequent in normal populations, that it's very unlikely that they are actually driving cancer, we also integrated this data from the NOMAD dataset. So in the end, every of the 5 million of coding mutations in COSMIC have this description, and you can see all these details about frequency across different cancer types. You can see its frequency in normal populations, and you can see all the annotations for ClinVar, as well as what is the result of the DNDS algorithm for this mutation. TP53 again. So this is about these 3 distinct peaks of recurrent mutations in TP53 that were quite surprising to be found there, because TP53 is a classic tumor suppressor gene. So we normally don't observe these things. COSMIC 3D allows us to project all the mutations in COSMIC on the 3D structures of the proteins. And it's a very nice tool to explain some mysterious cases like this one. So the first observation is that all 3 peaks are actually clustered in the 3D space. That's not obvious from the linear diagram, of course. It's impossible to see that. But on the 3D structure of protein, you can see that they are actually clustered around the sites that interact with the DNA. And 2 of these sites are directly attached to the DNA, and they are recurrent in cancer. What occurs is that these are in fact gain-of-function mutations. How they work is that they change the affinity, specific affinity of TP53 to DNA, and because of that, it doesn't bind to the right places on the DNA. The result is that instead of promoting apoptosis or generally protecting the cell from transformation, it reduces apoptosis and increases proliferation and invasiveness. Another interesting feature of COSMIC 3D is prediction of the impact of substitutions on the space occupied by amino acids. So we are not there quite yet that we can predict what is the impact of the structure of the whole protein, but locally we can map that. And here you can see that the wild type shown in pink occupies slightly lower, slightly smaller space than the most frequent mutant in EGFR. What goes further, we can identify the potential drug binding pockets on the 3D structure of the protein. And here, what you can see is that the wild type amino acid doesn't invade this pocket, while the substitution, the oncogenic substitution, is has bigger amino acid, and this amino acid invades the space. As a result, it prevents the drug from interacting with the protein, and that is the mechanism, molecular mechanism of the drug resistance. Another slide, I promise to get back. We can say something about how mutations are generated, but this is very basic. We also have something more sophisticated, and this can be applied to any next-gen sequencing sample. Basically, every sample has hundreds, thousands of mutations, and by integrating them all, you can produce this type of histogram called mutational profile. So basically, it takes into account the substitution itself, but also the neighboring nucleotides. What you can do next is use the convolution algorithms to identify patterns typical for different mutagenic factors. So As you saw, the tobacco smoke and burnt meat result in slightly different patterns of mutations. These mutational signatures have much higher resolution, and with these algorithms you can actually identify, if you can see in your cancer samples, if you can see patterns typical for tobacco smoke or for some other mutagen. And COSMIC collects all these signatures and all the information that we have so far. So proposed etiology, what mutagen can cause this pattern, but also as they occur, what can be the clinical significance for these patterns. And this is another resource that is being developed, but it's not experimental by any means. We are curating these mutational signatures in collaboration with people working on it, and we are collecting only the signatures that are already approved and can be used. To give you an example of how they already can be used, is the homologous recombination pathway. When it's deficient, it results with incapacity of the cell to repair DNA properly. As a consequence, you see some mutations gathering across time in that cell, and that results also with very specific mutational pattern, and this pattern can be detected with this system. And what differs it from you can just check if you have mutation in genes that are controlling the homologous recombination pathway, but in this case, you are checking the results of that. So no matter how this pathway is affected through mutations or through something else, you are able to detect the effect. And based on that, you can actually select the therapy that is targeted against deficiency in this pathway. So all these data sets in COSMIC, including these interpretation projects, can be downloaded from QIAGEN websites. And if you have any questions about that, please contact QIAGEN's support team. Also, please contact us if you have any questions, any requests. We are really happy to hear your feedback about anything, because we want to work closely with the community, and we want to stay relevant, and we want to be efficient in helping cancer patients.
Thank you again, Dr. Sondka, Ms. White, for this informative presentation. Again, audience members, if you have any questions, please email us at the email address listed here, or visit www.digitalinsights.qiagen.com for more information. Additionally, for a free download of COSMIC sample data, please visit the URL listed here. Thank you again, and have a lovely rest of your day. Until next time.